He's Dr. Jim Faber from University of North Carolina, and he's going to speak with us today on adverse effects of genetic background and risk factors on the collateral circulation. Jim?
Well, thank you very much for that introduction and also for the invitation to speak here. The organizers, I appreciate it very much. I'm going to talk about a type of vessel that we really haven't discussed at the meeting yet today, and these are collateral vessels. My laboratory is interested in collaterals in brain, in heart, and in peripheral limb, and what I'm going to confine my talk to and focus on is really collaterals within the brain. I'll say a few things about collaterals elsewhere, but mostly give you examples of what we've found in the brain. For those that are not familiar, have the lights down a little? Okay, thank you. This is a slide showing the coronary collaterals in an individual that's 48 years old, completely healthy. What's happened here is a balloon has been placed in this, the left anterior descending, and flow is occluded, and shortly thereafter, contrast is injected into the LAD, and you can see that it's swept around and retrogradely perfuses the tree whose trunk has been blocked, and that's because this individual has a substantial coronary collateral circulation, as evidenced by unusually large collaterals that can be imaged. Thank you. You can image these in this particular individual with X-ray angiography because they're unusually large. Collaterals generally can't be imaged directly. They're under 100 microns in diameter, but this is a good example of these. Obviously, they're very important vessels. They act as endogenous bypass vessels. If you have them in abundance, you have a substantial amount of protection if there's an occlusive event that occurs. They're very unique vessels. And I have shown this film many times, but it illustrates this purpose. See, are you able to turn down the lights? Okay. So this is a mouse anesthetized cranial window. We're injecting 1 micron microspheres that are fluorescently excitable so that they're as a flow tracer. And here's a little bit of anatomy to orient you. You can see the MCA tree is cross-connected to the ACA tree by the presence of a very tortuous vessel that's a characteristic of a collateral that is cross-connecting these. You'll also see that there are penetrating arterioles, just like the other branches of a cerebral artery tree in the pia, coming off this collateral and descending into the cortex. There are some venials in the background. So this is the flow that you see in these vessels in a sleeping anesthetized mouse. There is no pressure drop across them because there is no occlusive event. There is no flow in collaterals normally, and that's quite an unusual event in the vasculature. But that's the normal event or hemodynamic environment of collaterals. Oops. All right, thank you. Thank you. And so collaterals live in what I'd like to call is an at-risk environment, as you can see here. This is just showing a collateral that's cross-connecting the distalmost arterial branch of the ACA tree and the middle cerebral artery tree, MCA tree. And as I said, there's no net flow or very low flow in collaterals under normal resting conditions. So collaterals live in an environment of low and disturbed shear stress. They're also under substantially increased circumferential wall stress because all of the kinetic energy is converted to potential energy. As well, because there's low flow, there will tend to be lower oxygen levels you can predict. We haven't measured these in collaterals versus a distal arterial. Now, we think that and these kinds of conditions favor oxidative stress, as we know, inflammation, atherosclerosis, and the conduit circulation. In the microcirculation, these kind of conditions favor pruning events to remove these types of vessels. So we are finding, and these results are not published, that collaterals have some, we believe, adaptations to live in this environment. They have thicker smooth muscle walls, and you can see that on this collateral compared to a distal arterial of the same diameter. You can see, or we have found as well, that there's a higher level of certain markers that indicate proliferative activity within the vascular wall. Now, a couple are shown there. As well, there's a balance, it seems, elevation and expression of certain anti-proliferative, anti-inflammatory markers. And I point out eNOS, and we'll come back to that. There's also, compared to distal arterials, lower expression of VCAM and elevated expression of the anti-inflammatory transcription factors, KLF2 and KLF4. So one of the things I want to leave you with is that we believe collaterals in all of us are vascular canaries. They sit in an adverse environment, and they are susceptible to being lost. They have another unique characteristic. They're very sensitive, at least in the mouse, to genetic polymorphisms. And this shows you one species of mouse, the black 6 mice, that has abundant collaterals between the MCA, ACA, and PCA trees. And this is a BALB-C mouse, a albino mouse in this case, that has almost none, averages about 1 between these trees. And as you might predict, if you ligate the middle cerebral artery to simulate an acute stroke, they have a massive infarct compared to the black 6. The general arterial venous and lymphatic circulations are normal in these two species. This is the same in other tissues of these two mice. This is skeletal muscle shown here, and you can see the same events, so that the collateral extent in one tissue we find in species of mice reflects or predicts the collateral extent in other tissues. Same things you would have if we do hind limb ischemia experiments. You can imagine the BALB suffers much worse ischemia. We've done genetic studies to try to identify whether, because we have such a big phenotype in the BALB-C and black strains, we have done genetic studies not only in those, and Dr. D'Amato nicely explained to you the linkage analysis, or QTL. So we did QTL studies. We've done SNP association mapping against not just those two strains, but against 23 additional strains. And we've done congenic find mapping. And the result of this effort has been that we've identified a single locus in the mouse species, and at least in these 25 strains. We've named it determinant of collateral extent number 1. And it accounts for 85% of the variability, the difference in collateral abundance, stroke severity, that you see in the black and BALB, as well as in those other strains. These are the elements that are in this locus. There are 28 protein coding genes. We've got it down to 700 kb at this point. 27 SNPs are responsible for this, presumably. But what's surprising is none of the protein coding genes is an angiogenic gene. We're trying to look at each of these now. Knockouts for these particular genes have no effect on collateral abundance. As well, a deletion mutant, eliminating all of these, has no effect whatsoever, so that helps us. We'll be looking at the other two shown in green. We're using gene targeting and editing approaches to examine RAB-EP2, which is a very high priority candidate for us. And as well, we're trying to eliminate the high SNP region shown by that brown bracket as another approach using introgression of a large bacterial chromosome methodology. So can't tell you what this is yet, but we're working hotly on it. Well, others are interested in these variability, especially stroke neurologists, in this kind of variability we've shown in mice, and that is because they have found that cerebral collaterals are very important and vary widely in patients, acute stroke patients. This is observed when they identify a patient that has, for example, a stroke in the trunk of the M1, so a cerebral artery stroke. They can measure the collateral score, the retrograde perfusion of this tree, using neuroimaging techniques. And when they do this, many investigators have pointed this out, that there's a large variability. So here's a patient with an M1 occlusion on subacute ischemic stroke evaluation. And what one finds is that this person has very good retrograde perfusion of this tree. And the presumption is that they have abundant collateral circulation. And it turns out that about 20% of us or at least stroke patients, have very poor collateral status. This correlates with large initial infarct volume, greater infarct growth over time, poor response to thrombolytic therapy, increased risk and severity of hemorrhagic progression with treatment, worse functional outcome. There's evidence as well that there's this wide variation exists in other tissues, heart and hind limb or lower extremity. This has now been shown in healthy humans without coronary artery disease. So this locus, DCE1, we believe it harbors a major driver locus or a link in the collaterogenesis pathway, because these differences in mice are established late during embryogenesis, when the collateral circulation is laid down between arterial trees and tissues. We believe it might be a risk allele for collateral deficiency in brain and in other tissues. And so with this in mind, we have just begun a study in humans together with 10 stroke centers, genetic determinants of collateral status in stroke. These are patients with acute ischemic stroke. This is a SNP association mapping study across this human locus, which is on chromosome 16. And we're also looking at some related genes that we know are part of this collaterogenesis pathway. And the primary endpoint of that is collateral status as measured. We wonder whether we might be able to, from this information, obtain a molecular marker for genetic dependent differences in collateral extent. Okay, second thing that I wanted to talk to you about is that collateral's not only sensitive to genetic differences, but also environmental differences. And we all know that aging, we've heard plenty about this, cardiovascular risk factors increase the severity of stroke, of MI, acute MI, of peripheral artery disease, and as well that these risk factors, many of them hypertension, well documented, causes a decrease in the density of arterioles and capillaries, at least in some tissues. I'm just going to summarize what we've found. We have found that collaterals are lost progressively with aging. In, for example, the black 6 mice, both the loss of number and diameter of pio collaterals and collaterals in skeletal muscle. We see that this is massively accelerated in time in mice that are deficient in nitric oxide. They are losing their cerebral collaterals, much bigger strokes, hind limb collaterals, much worse injury there and ischemia by 3 months of age, rather than 16 months of age required otherwise. We see the same thing if we treat animals chronically with L-NAME and correct the small blood pressure increase that occurs with that agent and with eNOS knockout mice. Hypertension alone causes collateral loss, and I want to show you just a little data to demonstrate that. These are not published. This shows you that these are renin transgenic mice. They're heterozygous, have a 20 millimeter rise in blood pressure. And what you can see is that if you look over time, there's a progressive loss of pio collateral number in these mice. And we see the same thing for a decline in the diameter of those collaterals that remain. We also see that if you increase the blood pressure, and in this case we'll be comparing heterozygote to 1 copy of the transgene producing elevated renin versus 2 copies higher blood pressure. And you can see now that even in a that pio collateral number and diameter now are lost in even 4-month-old mice. The same thing is seen in the hind limb. And of course, we show in these animals worse stroke and worse hind limb ischemic injury. Mechanistically, we don't know by any means what's causing this, but we do find that collaterals in the hypertensive animals have higher basal proliferation. This is labeling with EDU over a 6-month period of this hypertension. We also find that the collaterals get longer, and that would be expected. Thank you. With elevated proliferation. So a hypothesis we have is that the chronically disturbed hemodynamic environment of collaterals places them at a higher proliferative potential, their endothelial cells in life, that this, if you add to it a disturbance like hypertension, that this can accelerate senescence of endothelial cells, leading to rarefaction. We have looked at these other models as well. This work will be submitted soon, and all of these risk factors cause collateral loss. So does smoking. We're looking at that right now. And it's important to point out that we don't see any effect on distal most arterioles in the cerebral circulation if we use those as a measure of collateral versus non-collateral 20-micron arterial. Common to all of these is, of course, endothelial dysfunction, vascular inflammation. I don't have much time left. I just want to share some very preliminary data where we've looked at Alzheimer's disease, and then I will stop. We asked the same question: does Alzheimer's potentially cause loss of collaterals? There's, of course, a lot of rationale for this. We've heard at this meeting that Alzheimer's disease involves a deposition of amyloid on pale blood vessels. And this happens actually before you start seeing in some of these models, neuronal amyloid deposition. I'll skip some of those right there, but this is our attempts to try to ask this question, and this is a triple transgenic mouse, Alzheimer's disease mouse model, and you can see the penetrating arterioles going down from the MCA there. This is a very early stage in this disease. It's known there's no neuronal plaque yet showing at this stage, but what we find is, indeed, there's a loss of collateral number, diameter tends that way. The vessels are longer. The resistance can be calculated to be higher. We haven't confirmed that elevates stroke, but we expect it. And once again, no effect on distal arterial number or diameter, penetrating arterioles coming off of them. No effect at 1 month of age in these AD mice. We have much to do here, but we wonder if these effects might contribute to the known elevated stroke severity in AD patients and VCI patients, and of course, whether it might have effects on cognitive impairment. I'm going to skip last concepts about that possibility in rats and humans as well, and finish with some lessons. First, that collaterals are very unique vessels. They are both in the way they form, and I haven't had time to tell you about that, but quite different than embryonic formation of the other vessels. Their anatomy is different. They're very tortuous, for example. They sit in a strange place. Function, their expression pattern appears to be different in the limited studies we've done so far. They live in a special hemodynamic environment. We believe they're kind of canaries of the circulation. They have high susceptibility to genetic polymorphisms and environmental, if you will, stressors. Some gaps in the field. Obviously, we need to identify this pathway that DCE1 is critically involved in, and we think it's impacting these effects at the level of late gestationally in the embryo. We'd like to know whether or not this collaterogenesis pathway could inform us about how we might induce new collaterals to form in adults with ischemic disease, in both in acute MI especially. We'd like to know about the mechanism of rarefaction, and there's much to be done by many people on trying to understand this, and can we slow or prevent it? And we're doing experiments now in that realm to test that approach. Can we identify markers for collaterals that might be used? And finally, what is the role of rarefaction? Is there a role for rarefaction in cognitive sequelae that occur in AD and VCI? So with that, thank you. I certainly want to thank the laboratory and all of the people that have done this work in the lab, and 3 of these investigators, Dr. Zhang, Lucidi, and Jin, are here with me in the audience. Thank you very much.
Thank you, Dr. Faber. That was terrific. We have time for about 2 questions, very briefly.
Very nice talk and very interesting ideas. So, these collaterals are, if they're ischemic dependent somehow, did you look at HIF signaling? That's sort of one question. Are these collaterals decorated to a greater extent with VEGF receptor 2 or?
VEGF receptor 2 is higher, yes, it's expression. That's just messenger RNA and isolated vessels, nanostring panel.
I think
VEGF's expression is not elevated, but receptor 2 is.
But if the receptor is elevated, that I think is very important. And also, did you look at markers of arteriogenesis? Because they may also be upregulated. But it seems to me this VEGFR2 expression may be a very good explanation.
Yes, we know VEGF is required for forming these vessels late embryonically. Those studies, we've published a couple of papers on that. If you knock down or knock out VEGF or knock it up, you make fewer or more collaterals in the embryo. They persist to adult.
HIF signaling.
Yes. Thank you.
You have
Jim,
the last
that-
question.
That was a very nice, interesting talk. I was wondering, since these vessels are in such a special environment, are they prone to collect inflammatory cells? Are they potential sites for metastasis if you have a tumor cell?
Now, those are very interesting questions. We're just now looking into, in these models that I've been showing you, especially the AD model, whether there is a special accumulation of monocytic cells or microglia that end up migrating to there from the brain itself. Can't answer that, but those are very interesting questions.
Thank you. We'll have to move on. Thanks.